Hello friends, welcome to BMH Learning. This video is about Catalase test. The test demonstrates the presence of catalase. Catalase is an enzyme which catalyzes the release of oxygen from hydrogen peroxide. So mainly the catalase test is used to differentiate catalase producing bacteria such as Staphylococci from non-catalase producing bacteria like Streptococci. Generally, in routine culture, 3% hydrogen peroxide is used. But for detection of catalase in anaerobes, 15% hydrogen peroxide is used. Principle: the enzyme catalase is responsible for the breakdown of hydrogen peroxide into oxygen and water. The principle is based on the evolution of oxygen once the peroxide molecule is broken. So when a bacterial inoculum is treated with hydrogen peroxide, either there would be rapid elaboration of oxygen bubbles or absence of oxygen bubbles. Oxygen bubble formation indicates the presence of catalase in the bacterial sample, whereas the absence of oxygen bubbles points to the lack of catalase in the bacteria. So this way, the bacterial species are differentiated from each other. Keep one thing in mind, that the bacterial culture should not be more than 24 hours old. Procedure: now there are two methods by which the experiment can be performed. First is the tube method. First of all, take two test tubes containing 1 to 2 ml of hydrogen peroxide solution. With the help of a wooden stick or glass rod, take several colonies of the test organism. We are using two different bacterial species here. Now, immerse the stick having bacterial colonies in separate test tubes containing hydrogen peroxide solution. You can notice in one bacterial species there is immediate bubbling, which shows the bacterial sample is possessing catalase enzyme. In the second case, no bubbling is observed, which demonstrates the bacterial species does not have catalase. Second is the slide method. Like in the previous method, here again we use a sterile stick to transfer several colonies on the surface of a clean, dry glass slide. Once the bacterial growth is over the slide, a drop of 3% hydrogen peroxide on the glass slide is placed. Observe for the evolution of oxygen bubbles. These are some of the examples of both catalase and non-catalase producing bacteria. Now there are many uses of catalase test. Morphologically similar Enterococcus and Staphylococcus can be differentiated. The test is also valuable in differentiating aerobic and obligate anaerobic bacteria. The catalase test is generally used to differentiate aerotolerant strains of Clostridium from baseless species. This was all. Thanks for watching.